Hi dear students, a warm good morning to all of you. Welcome to one more session of immunology, and today we're going to talk about the antigen antibody reactions. We all know that within our body we come across or we get exposed to different antigens, and these antigens need to be killed. And one part of our immune system is the production of antibodies, and these antibodies try to interact with the antigens, resulting in antigen antibody reactions in trying to kill these antigens or neutralize these antigens. This system is referred to as the humoral system of the immune system, and today we're going to talk about the antigen antibody reactions. Antigen antibody reactions, of course, do happen within the body. And when it happens within the body, it is referred to as in vivo, and they provide protection against pathogens. But at the same time, these antigen antibody reactions might be carried out in vitro, in the lab, or in a diagnostic center uh, in order to find out the presence of a particular infection within your body. And in such cases, it is called the serological reactions. Now, what is the purpose of these reactions? They aid in the disease diagnosis, sometimes to find out the humoral antibody level, to detect the presence of some antigens, and also for the detection of hormones or enzymes. So, though we say that antigen antibody reactions happen within a body as a protective mechanism naturally, the same antigen antibody reaction can be used for a diagnostic purpose to find out whether you are infected or not, and also to find out your immunity level. Now, how will we detect your immunity level? You might have a doubt like that. I have told you like the antibodies formed in a sequence M, D, G, E, and A. So if you have a IgM type of antibody within your body, it indicates that it is a recent infection. But if you have infections on the other side, like the antibodies of other classes, it indicates that the infection has already been proceeded. So, by analyzing which type of antibody you have within your body against a particular antigen, you will be able to identify what is the stage of your particular disease. Okay, this is now in the case of almost all diseases, you can use this strategy. And also for the diagnosis, that is whether you are infected or not. For that, you will check the presence of antibodies within your serum. I hope you know what is the serum. Serum is coming the portion of, we have already discussed that the serum is a portion of the blood excluding the RBC. That is, when once you take your blood, centrifuge it, the RBC gets settled down, and the liquid portion of the blood, which is found as a supernatant, excluding the RBC and all that, that will constitute the serum. And within the serum is present the antibodies. So when you take antigen antibody reactions in vitro in a lab or in a diagnostics center with the aim of various purposes, such reactions are called the serological reactions. Now, antigen antibody reactions also do have some features. They include that they are specific. It will include a entire molecule, and denaturation does not happen for the antigen and the antibody. And sometimes the combinations with surface antigens happen, and it is reversible. And of course, the strength as well as the strength of the affinity and avidity of the antigen antibody reactions are also being present. Let's go into the details of these salient features. Now, what do you mean by specificity? We know that a lock and a key are being found to be specific. If you remove the key of a specific lock, and use something else, it will not work. The same way here, antibodies are being produced against specific antigens. You can see that at the antigen binding site, okay, the, that is mainly at the hypervariable regions. At that particular region, you can see that the antibody becomes specific for a particular antigen. Now here in this picture, you can see that only the yellowish antigen can come and bind to the antibody. If you try to put the violet, red or the green antibody to come and bind with the antibody, it will not happen because antibodies are being produced specific to a particular antigen. So that's the meaning of the specificity of the antigen antibody reaction. Now coming to reversibility. Sometimes you can see that the reactions can be reversible. Irreversible means after binding, the binding can be moved back, or it might be nullified, or it might, like, the situation of binding can be reversed to non-binding. And this reversibility of the antigen antibody reactions is mainly due to the presence of non-covalent bonds such as the hydrogen bonds, electrostatic bonds, van der Waals forces, and the hydrophobic bonds. And you can see multiple bonding between the antigen and the antibody ensures that the antigen will be bound tightly to the antibody. So all these are coming under the features. And you can see in the second feature that the entire molecule will react. That is the antigen antibody reaction. In during that, what happens? The epitope as well as the part of the epitope as well as the antibody will react as an entire molecules, and there will not be any denaturation of the antigen or the antibody. And you can see that if you consider a whole antigen, the binding of the antibody to the antigen mainly happens within the what? Surface antigen of the surface antigens. Now, what do you mean by surface antigens? Now, consider a pathogen. Okay. Now, I'll give you an example. If you have COVID-19, you have lots of spike proteins on it. That is a surface antigen. So, the binding of the antibody mainly happens more on the what? Surface antigens, or those antigens which are being found on the surface of the pathogen. So that is also a feature of the antigen antibody reactions. So you understood what is specific. What do you mean by the entire molecule isn't reacting? There is no denaturation, and surface antigens are being binding, and the reaction has been found to be reversible. And now let's explore the features of affinity and avidity. Now, affinity, it refers mainly to the strength of the reaction between a single antigenic determinant and a single combining site on a antibody. And some of the attractive as well as the repulsive forces operating between the antigenic determinant and the combining site of the antibody is also being referred to as affinity. So the strength of the reaction, that's what we have wrote. Now consider this case. In the first figure, you can see that the epitope as well as the paratope, they have a high affinity because they fit in properly. But coming in the second case, you can see that there is a difference in the structure over here, and so they have a low affinity. So the strength of the interactions between the antigenic determinant and the combining antibody. Now here AB refers towards the antibody. The strength of binding of this is referred by the term affinity. Now coming to the avidity. It mainly involves the measure of the overall strength of binding of an antigen with many antigenic determinants and multivalent antibodies. Now, in the previous one, you were considering the strength of the reaction between one single antigenic determinant and the antibody. But in the case of avidity, it is just the strength of binding of an antigen with many antigenic determinants and a multivalent antibody. As you can see over here, okay, this is affinity, where each antibody or each epitope is binding with the paratope of the antibody. But in avidity, it mainly includes the overall strength of binding of an antigen with many antigen, like the antibody with many antigenic determinants and multivalent antibodies. And avidity is influenced by both the valency of the antibody and the valency of the antigen. Avidity is more than the sum of the sum of the individual affinities, and affinity refers to the strength of binding of a single antigenic determinant and an individual antibody combining site, whereas avidity refers to the overall strength of binding between a multivalent antigen and the antibody. So just remember, when it comes to avidity, we refer to as multiple antigenic determinants are reacting with a multivalent antibody. Either a multivalent antibody or it could be different antibodies also as such. So I told you about the features. Now, what does or how does this antigen antibody reaction happens? Antigen antibody reactions can occur in different forms, and based upon that, we classify them into different cases. We know that sometimes the antigens which we find will be of different types. Sometimes you can see that some antigens will be soluble antigens, that is, they will be cell free, some molecules or something like that. Or sometimes there would be viruses which are been forming. Or sometimes you can see that they would be surface antigens. So the antigen antibody reaction can happen in different ways. Sometimes you can see that when the soluble antigens are being formed within your body, okay, that is cell-free molecules in solution are being formed. That is from the surface of the pathogen itself, some proteins might be coming out and they might be soluble within your body. In such cases, the antigen will react with the what? Antibody, and it will result in a type of reaction called precipitation. And sometimes when antigens like the viruses come, the antibodies will come and bind across the virus and prevent the binding of this virus onto your cell. That is called the process of neutralization. Okay, and sometimes once the antigen is being formed, okay, the what happens, the antibody also comes and bind with the pathogen, and certain other molecules called the complements also come and attach, that is onto the antigen antibody complexes. Various other proteins called the complements which would aid in the lysis of the bacterial cell, okay, and a complement fixation reaction can happen. And sometimes you can also see that if there is a particulate antigen, you know what's a particulate? It's particulate means something which is solid. It's like a particle. It's not the soluble like cells. The pathogen as such is no particulate one. It's not a soluble one. In such cases, what happens? You can have a agglutination reaction. So if the pathogen is a soluble antigen, it will go for a precipitation reaction, and if it is a particulate antigen, it will undergo a agglutination reaction. And sometimes what happens, you can see antibody is also involved in opsonization. Now what is opsonization? Opsonization is a process of increased phagocytosis. So once the antigen is being coated on the once the antibody is being coated on the antigen, you can see that the antigen antibody complex might be engulfed by the macrophage, and the increased phagocytosis can happen. And that is a process of opsonization. So the antigen antibody reactions within your body can happen in different ways depending upon the type of things with which it interact. If it is a soluble antigen with which the antibody interacts, it will result into a precipitation reaction. If it is a particulate antigen, it will result in the formation of agglutination. And if it is a virus, it could bind with an antibody, causing blocking the virus binding to the cell, and that is called the neutralization reaction. And if an antigen antibody complex along with a complement is been attaching, it will cause the that is called the complement fixation, and it will cause the lysis of the bacterial cell. And sometimes you can see that once the antigen antibody complex is being bound, macrophages, which are phagocytic cells, might come and attach onto these antigen antibody complex and engulf and take it in, or increase the phagocytic rate of the what antigen antibody complex along with certain other proteins called the opsonins. And in such cases, that is referred to as the process of opsonization. So these are the different types of the antigen antibody reactions. And this is just another depiction of what I mentioned earlier: the neutralization reaction, the agglutination reaction where particulate antigens are being formed, the precipitation reaction where soluble antigens are being formed, and the complement fixation action. Okay, so these are the different steps. I had already mentioned it. So what is it? Agglutination: the antibodies clumps the antigen together, which are later destroyed by the phagocytes. Thus, by clumping them together, the phagocytes can detect them more easily. So, the antigens will go and bind onto the what? The antibodies will go and bind onto the particulate antigens over here, and so a clumping will be formed. And precipitation reaction, the antibody is getting bind with the what soluble antigen. And in the case of opsonization, antibodies are coated on a microbial surface, after which the antigen locks in, and this also makes it more susceptible to phagocytosis. And neutralization is a process in which the antibody blocks or neutralizes the harmful chemicals produced by the antigens. Also in the case, you can also find viruses also will be destroyed by this, will undergo a process of neutralization. And complement activation: once the lock and key mechanism perfectly fits into the place, it will lead to the cell lysis. Of course, complement activation will involve the production of complement proteins also. So this is the main features, types and steps of the antigen antibody reactions. And you can have visual effects can be divided as a primary stage, the secondary stage, and the tertiary stage. In the primary stage, if the reaction has been happening, you might not find any visible effect, and it is reversible because lots of forces such as the van der Waals forces, ionic bonds, and hydrogen bonds are being involved. And in the second stage, it could be you will find demonstrable events. Now, in the case of precipitation and agglutination and the lysis and killing of cells, in these are demonstrable events. And the tertiary stage, it happens mainly in the these it is in the in vivo. You can see that the neutralization as well as the destruction of injurious antigen as well as tissue damage can result as a part of the antigen antibody reactions. So with this, I'm think you got an overview of the antigen antibody reactions. We would be dealing with more details of the antigen antibody reactions in the coming classes. Thank you for now.